dear learners, welcome to Learn Microbiology. In this video, we are going to study about the negative control of transcription in prokaryotes repression. First, let us understand what do you mean by negative control and what is the meaning of repression. In this video, we are going to understand this repression pattern by using an example of Arginine Operon. So let's get started. Negative control of transcription, it is a control mechanism that prevents the transcription. Students, in the previous video, we have already learned what is the importance of regulation of transcription. If you have missed it, do check out the link in the description box. Now, there are two types of control of transcription. First type is called as the positive control, wherein your transcription is positively regulated, and second term is the negative control, wherein your transcription is prevented. We have learned already that why sometimes the cell decides to prevent transcription. It is because when the products are not required and the cell goes on transcribing and translating the genes and producing the gene products, then it goes for waste of resources and energy, right? So hence there is a mechanism called as negative control. In this mechanism, there are two types. First one is repression and second one is induction. So in today's video, we will study about repression, and in the next video, we will learn about the induction. So let us understand what is the meaning of repression and how transcription is negatively regulated by repression. So this is the statement which help us to understand about repression. Let me read it for you. The enzymes that catalyze the synthesis of specific product. We all know that for the synthesis of any products in a cell, the cell uses enzymes, correct? So these enzymes are not made if the product is already present in the medium in sufficient amounts. Let us understand what this means. For example, we have an enzyme E which is involved in the synthesis of our product P. Okay, let's take it as an example. Now, when this product is already present in the media in sufficient amount, then will the cell require the enzyme? No, right? Enzymes will be required so that it can produce the product P only when P is not there in the medium. When you give P in the medium, when this product is already present in sufficient amounts in the medium, then these enzymes are not made. Let's revise how enzymes are made. Can you recollect it from the last video? How gene expression takes place? There are genes which code for enzymes. Translation of the mRNA, which is made by the transcription process from a gene, gives us the enzyme. So basically in this mechanism, if the product is already present in the medium, ready-made, then there is no need of the enzyme which is required to make that product. Yes or no? Correct. So what will happen? The transcription of those genes are repressed or it is blocked. Fine. Let us understand this with one example. Let's take the example of arginine, which is an amino acid. Now this arginine amino acid, if it is already present in the media, then the enzymes which is needed to prepare this arginine amino acid is not required. Yes or no? In that case, the genes which code for these enzymes needs to be blocked to prevent energy and resources waste. Correct. We will understand this with the help of Arginine, Operon in this video along with an animation. So watch the entire video. So that is the sentence. Next sentence is the one which I have already mentioned, that the enzyme which are needed to synthesize this arginine, they will not be made when it is present in the medium, and it will be made only when arginine is absent from the culture medium. If you don't give arginine ready-made to the bacteria in the medium, only then it will decide that it has to synthesize these enzymes so that it can prepare arginine by itself. Okay. So hence here what we can say is, if arginine is present, then it is preventing the synthesis of these enzymes, correct, which are needed to prepare arginine. Hence, this arginine, it is going to act as a co-repressor. Okay, it is going to act as a co-repressor and it is going to repress its own synthesis. Okay, this is called as enzyme repression. So I hope you might have understood what is the meaning of repression. So let's go for a quick revision. So what is repression? The end product, what it does, if it is already present, readily available in the media, then it will block its synthesis of the enzymes which are needed to make that product. This is called as enzyme repression. Now let's take the example of Arginine Operon. Let's take a scenario. Here you can find a culture media in which bacteria is growing without arginine. So remember, in this media you have not provided any arginine. So can you guess, will the bacteria prepare arginine now? Definitely yes. Why? Because it is not having arginine readily available in the media. So it has to prepare the arginine itself. So now those genes will be active. Transcription and translation of their genes will take place. And the enzymes needed to prepare this arginine will be synthesized. Right? So let's take a experiment. Let's do an experiment here, and that experiment's results will be plotted in a graph, and this is the graph. As you can see, on the x-axis we are having time, so we are going to see this. We are going to record the product formed with respect to time. So we also have a timer on this left side, correct? Then let's take the relative increase on the y-axis. So basically in this experiment, we are going to monitor the amount of 3 components. Okay. So what is happening here? Bacteria is growing in a media without arginine. So when this bacteria is growing, we are going to start our timer and we are going to find out the concentration of 3 components. What are the 3? The first one is cell number. Cell number means the number of cells. So as the bacteria is growing, the number of cells need to increase. Okay. So with time, we can find that number of cells will go on increasing. Second, we will also monitor the total proteins. Like bacteria not only make enzyme which is needed for the synthesis of arginine, it is also going to prepare n number of enzymes and n number of proteins. So we are going to monitor the total protein content also. Third, we will monitor the important component, that is the arginine biosynthesis enzymes. Now why we are monitoring this? Because arginine is absent. Okay, read it here properly. Arginine, you can find it, this media is without arginine. So when this arginine is absent, then the bacteria needs to prepare arginine biosynthesis enzyme. So with time, as the bacteria go on dividing and its cell number go on increasing, you should also expect that total proteins will increase. Also, the arginine biosynthesis enzymes concentration will also increase. Okay, so let's start the timer and plot the increase in this graph. So as you can see here, bacteria started to grow, and you can find with the passage of time, the cell number, the graph which is the first one in blue color, that is your cell number, it goes on increasing. The second graph which represents total protein, it is also increasing, as well as arginine biosynthesis enzyme is also produced, hence the graph is also increasing. Now we are going to put a twist to the experiment. Okay, what we are going to do is, after some point of time, we are going to look properly. We are going to add arginine. Okay, and what we did was, this media was not having arginine, correct? So the bacteria was synthesizing this arginine biosynthesis enzymes. Hence, its concentration was increasing, as we can see in the third graph. But what we did after some time, we added arginine in this media. Now let's again start the timer and see what will happen to the cell number, total protein content, as well as to the arginine biosynthesis enzymes concentration. So let's do it. So timer has started, bacteria is also growing. Stop. Now you can see there is a different pattern in the graph obtained. Cell number keep on increasing even after you add arginine. Similarly, total proteins is also increasing. But you can see here that the concentration of arginine biosynthesis enzyme went to a constant state. It is not increasing. So over here, when you add arginine, immediately the synthesis of this arginine biosynthesis enzymes had stopped, because now arginine is readily available in the media. So now the cell will not take the effort to prepare these enzymes. Correct? So that is why we can see the concentration of these enzymes go for a decline. Correct? But can you see the total proteins? The other proteins, they are not affected. Only arginine is affected. Arginine synthesis enzymes are affected. It means arginine is acting as a repressor, or the word or the term which we use is called as co-repressor. Correct? And it is going to prevent its own synthesis. Okay. So we have understood this concept with the help of this experiment. Now, you might wonder what exactly happened after arginine was added. How the cell came to know that arginine is there in the medium. Now it has to stop the transcription of those genes, isn't it? So let's understand what happened that prevented the synthesis of this arginine biosynthesis enzymes in the next slide with an animation. So watch the entire video. Okay, let's move here. Now again here what we will do is, we will take a flask where bacteria is growing. Now this bacteria is growing in a media without arginine. There is no arginine in the media. Correct? So can you guess? The cell needs to prepare these enzymes needed for synthesis of arginine. Yes or no? The answer will be yes. It has to prepare those enzymes. So let's have a look at the arginine operon. So this is our arginine operon. As we learned in the previous video, that operon has a promoter sequence, which is the driver. Then it has an operator sequence. Then there are three structural genes. You can see here arginine C, arginine B and arginine H. So basically these three genes are going to code for three different enzymes which are needed to synthesize arginine. Why? Because now arginine is absent in this media. Okay. So what else is there in an operon? We have already learned we require an RNA polymerase enzyme which is needed for the transcription process. Then don't forget the repressor protein, because this repressor is going to bind at the operator site. Then what will happen? Everything is ready, all set. You can see the repressor is not bound to the operator. Why? Because there is something lacking here. You can see there is something lacking here. What is lacking? A co-repressor. Only when the co-repressor bind to the repressor, it will get active and it can bind to the operator and thereby prevent transcription of these structural genes. But now co-repressor is absent. Who is the co-repressor? It is arginine itself. So arginine is absent. Hence the repressor is in the inactive state. It cannot bind to the operator. So now the path is clear for RNA polymerase. So it is going to move further and initiate the transcription of the 3 genes C, B, H. And as you saw, we can see a polycistronic mRNA is getting generated from this, which is corresponding to these 3 structural genes. Now what is the next process? The next process is translation. So in the translation, what happens? Yes, we are going to get the 3 different proteins. That is the 3 different enzymes which have been encoded by this gene 1, the gene 2 and gene 3. So now these 3 biosynthesis enzymes are being produced. Correct? So this is the number 1, this is number 2, this is number 3. Now these 3 enzymes are going to synthesize what? Arginine. So when arginine was absent in the media, there was no co-repressor to bind to the repressor, so that transcription was not prevented. But RNA polymerase initiated the transcription, and you got these 3 enzymes, and these 3 enzymes, they produce your arginine. Correct? But what will happen if the bacteria is growing in a media containing arginine? How arginine is going to block the transcription? Are you wondering? Let's also understand that with an animation in the next slide. So now our bacteria is growing in the media which is already having arginine, correct? So arginine, which is also called as our co-repressor, is already present in the media. So let's again take the example of our arginine operon with promoter, operator, as well as 3 structural genes C, B and H. So as we know, arginine is already present in the media. So arginine is readily available inside the cells of the bacterial cells. Okay. So we can see RNA polymerase very happily bound to the promoter and was ready to transcribe. We also have our repressor now, but we also have the co-repressor, right? So what happens? Our arginine, which is a co-repressor, it will now bind to the repressor. So once the co-repressor bound to the repressor, our repressor is active. Once the repressor is active, it will go and bind to the operator site. So once the repressor bound to the operator site, our RNA polymerase is prevented from initiating transcription. So as we can see, transcription is blocked. Correct? Thus we can see that arginine, which is the product, when it is present, it itself is functioning as a co-repressor, and binding to the repressor, it has blocked its own synthesis. Thus we can see arginine biosynthesis enzymes are not synthesized. So our co-repressor, which is arginine, has blocked its own transcription. So transcription here is blocked. So when transcription is blocked, this is an example of negative control. And since our end product is blocking transcription or it is repressing transcription, it is called as repression. So I hope you have understood what is enzyme repression and all your doubts have been solved. In case you have any doubts regarding this topic, do let me know in the comment section. So thank you all my dear students for watching this video. If this video was helpful, don't forget to like the video, share it to your friends and subscribe to Learn Microbiology.